The next speaker of today is Noemi Montobbio from the Istituto Italiano di Tecnologia, and she will speak about emergence of Lie symmetries in functional architectures learned by convolutional neural networks. Okay, you have the mic.
Thanks. So first, thanks a lot for inviting me. Compared to the previous speakers, I'm going to give a slightly different point of view. So I'm not focusing on the math in the formulation or training of a neural network in this case, but rather on the approximate geometric invariances that are found in the learned weights, so in the configuration that the network learns from natural images. So, okay, so first, just an outline of the talk. CNNs, that stands for Convolutional Neural Networks, as all of you probably already know, are a particular kind of deep learning architecture designed in analogy with information processing in the early visual pathway. So besides the typical hierarchical organization that we find in deep neural networks, we also have translation invariance that is enforced by local windows shifting over the spatial domain. So when I say early visual pathway, I'm referring to roughly all those processing operations that happen in the brain areas going from the retina to subcortical areas that are essentially the ones in the inside of the brain to the first areas in the visual cortex that receive and process the stimulus. So among these, the most studied is the primary visual cortex that I will call V1 for the rest of the talk. So a typical and also convenient way to describe the way that these areas process the visual stimulus is by means of group invariances. So I don't understand if someone is talking or no. Okay, so we can think of the image given as an input to a CNN as basically the projection of our visual field onto the retinal plane. And on the other hand, the first layer of filtering that happens in a CNN is typically associated with the processing happening in V1. In fact, in previous work, it has been shown that the filters of the first layer of a CNN trained on a set of natural images share some important geometric properties with receptive profiles found in V1. So essentially their orientation selectivity. So we can see immediately that there is no analogous in a CNN for subcortical regions, and also all the connections in a CNN are feed-forward, meaning that they map from one layer to the next. So the interactions between units belonging to the same layer are not modeled in any way in a classical CNN architecture. So the idea is that if we insert suitable computational blocks in a CNN that are compatible with this sort of biological mechanisms, then after training onto natural images for some task, for instance, classification, we could observe the emergence of geometric symmetries that are biologically relevant. So first, I'm going to show that inserting a simple pre-filtering stage before the first convolutional layer leads to radial symmetry, a radially symmetric pattern that is consistent with subcortical processing, so before the primary visual cortex. And second, lateral connections that are learned and that act on the feature space of the first network layer are going to lead to a connectivity kernel that shares some geometric properties with well-known properties of lateral connections happening in V1. So I hope the general logic is clear. So I just want to set a few notations and terminologies. So first, receptive field, that is the domain of the retina to which a visual neuron is connected. So basically the set of points to which a visual neuron responds. And this response, the set of points in the retina. And this response can be of different type and intensity. And this can be described by means of a function called the receptive profile. This function psi that is defined onto the receptive field. So importantly, this just describes the response to punctual stimuli that is just basically a luminous point or a Dirac delta defined onto the retinal plane. And that is also called the impulse response of the neuron. And that is going to be positive for excitatory response, negative for inhibition, and it's in the intensity of the response is expressed by basically the absolute value of this function. So the impulse response can also be written simply in terms of the evaluation functional this way. So actually, there are classes of cells in the visual pathways that are shown to behave like a linear functional onto a visual stimulus, so that from the impulse response, we can actually infer the response of the cell to any visual stimulus that we can model as a function defined onto the retina, so that this integral can be written not only for punctual for functions that are deltas, but for any function in L2 of the retinal plane, so that we obtain a linear filtering that is simply the integral of the profile, of the receptive profile of the cell to against the image I. So this linearity property is found, for instance, in the lateral geniculate nucleus, that is the most important subcortical structure of the visual pathways. So these kind of cells have a receptive profile that is approximately radially symmetric, so with positive values in the middle surrounded by negative values or vice versa. And this is typically modeled as a Laplacian of Gaussian function, so basically a Mexican heart function in 2 dimensions. And another important family of cells that have this property of linearity with respect to the stimulus are simple cells in V1. So these are also the first cells in the visual pathways that are selective to orientation. So their profiles are given by an alternance of positive and negative lobes that are elongated along a certain orientation that is the preferred orientation of the cell. So the most simplified model for the whole family of receptive profiles of these cells in V1 is given by a family of filters that is invariant by translations and rotations. So the family is indexed by the group of bidimensional translations and rotations, that is R2 times S1. Being a product space, that means that for each point of the retinal plane, we have a whole fiber of filters sensitive to all orientations. So, as I said, the response of such a cell to a stimulus I is expressed as a linear filtering this way. So if we want to describe the collective response of the whole bank of filters to a certain image, that can be written as a function onto R2 times S1. So for each point x, y, theta, we have the response of the corresponding profile to the image I. So this basically means that we are remapping the image I from the original positional coordinates to a new set of coordinates that are in a feature space that in this case encodes position times orientation. So we are modeling V1 as a feature space, a space of features that are not necessarily just position. So when so the phrase feed-forward connections in this case means such a functional that remaps the input from a space to a different set of coordinates. Whereas when I talk about lateral connections, I refer to those interactions that happen within the same brain area in biology and in terms of feature spaces that can be written as a kernel that expresses the pairwise interactions between points of the feature space. So for every 2 points, the kernel K gives the value of the strength and sign of the interaction between these 2 points. So in view 1, some interesting geometric properties are observed in the lateral connections as a function of the orientations. So as a function of the positions and orientations. So short-range connections. So short-range connections mean those connections, those interactions that happen between profiles that share the same positional coordinates but are sensitive to different orientations. So for these kind of connections, we observe a phenomenon that is called no-maximal suppression, meaning that the most activated orientation-selective cell tends to suppress the activations of other cells that are not optimally oriented with respect to the visual stimulus, giving a sort of Mexican hut shaped connectivity. Whereas long-range connections refer to those connections happening between cells that do not share the same receptive field, so that are connected to different areas of the retina. So in this case, the connections departing from a cell that is sensitive to a specific orientation have been shown to spread preferentially along the axis that is aligned with this orientation, and also they tend to target preferentially those cells that are either aligned or forming an arc of circle with this orientation. So this properties, that is anatomical property, that is anatomical, is very important since it's been shown to be at the basis of our, it's thought to be at the basis of our ability to complete partially visible contour. For instance, if we see a dashed line, we can still perceive it as a line. Okay, so how do we translate all of this in a CNN? So each convolutional layer of a CNN, each of the convolutional layers of a CNN performs a sort of linear filtering operation like the one that I was showing before. And importantly here, the filters are imposed to be invariant by translations, meaning that the filter shifts over the image and filters the image at each point, so that this linear filtering operation becomes a convolution. Now, as I was anticipating, it has been shown that the filters of the first layer of a CNN that is trained on a set of natural images, for instance, for classification, develop orientation selectivity, so that an important implication of this is that if we can associate each filter with an orientation theta, this means that we can, in principle, perform a change of coordinates in the feature space. So the feature space has two spatial dimensions and a third dimension that is just an index, right? It's the index of the corresponding filter to be learned. So if we can assign this index an orientation, that means that we can, in principle, rearrange the third dimension of the space to give it a geometrical meaning. So I'll go back on this later. So as I was saying, we want to insert a pre-filtering layer that is just another convolutional layer before the first layer of the network, but a convolutional layer with just one single filter. So thanks to translation invariance, this means a set of filters all with the same shape, but at different positions onto the image. So I'm going to compare the learned filter in this layer with the geometric properties of subcortical processing, since this happened between the input image and the first layer filtering. And second, lateral connections acting onto the feature space of the first network layer. So these connections act by modifying the activation of the layer by taking the average between the original activation and the activation updated by convolution with the connectivity kernel, as the one that I was mentioning before, but except that this kernel is learned by the network. So this acts on the feature space of the first network layer that is associated with the processing happening in V1. So we want to compare the geometry, the connectivity induced by this kernel with the properties of the lateral connectivity of V1. So those properties that I was describing before. So so we considered a very simple convolutional architecture that we trained for classification of a grayscale version of Cypher10, that is a dataset of low resolution natural images with 10 classes such as cat, dog, truck, and so on. So I'm not going into detail about the training, but I can, of course, if anyone has questions. Just wanted to mention this plot that shows the mean testing accuracy as a function of the number of convolutional layers. And the three curves are, so we see in blue a standard CNN, in orange a standard CNN with the pre-filtering layer, and in yellow the same with also the lateral connections. So we can see that the performance of the network is improved by adding these layers. And second, I wanted to say that we wanted to keep it simple. So we limited the number of convolutional layers to just 10, since we were not aiming at reaching state-of-the-art performances, but rather at the interpretability of the architecture. So in this case, we selected the model that reached around 80% of accuracy on the testing set. Now, unsurprisingly, a convolutional layer with just one single filter develops a radially symmetric shape, so no orientation selectivity. And this shape is closely fit by a Laplacian of Gaussian kind of profile that, as I was saying before, is an established model for receptive profiles in the LGN. So this mimics subcortical processing. But the nice thing about this pre-filtering stage is mostly in the effect that it has on first layer filters. Since in classical CNN architectures, basically the subcortical and early cortical processing are incorporated into a single layer. And so typically what we get in the learned filters of the first layer is a majority of orientation-selective filters plus a bunch of non-oriented filters showing for instance, a Gaussian-like shape. Whereas in this case, inserting these pre-filtering layers, a layer allows us to sort of decompose this first part of the processing into two mechanisms that are well separated and roughly corresponding to subcortical and early cortical processing. So we get a whole layer of orientation-selective filters in the first layer. So a way to extract the orientation, the preferred orientation of each of these filters is just fit each of them with a Gabor function. So basically a wave-like function that is modulated by a, a bidimensional Gaussian function. And so this allows us, as I was anticipating before, to change the coordinates of the feature space. So for each filter of the first layer, psi f, we can assign two positional coordinates, x and y, that are the coordinates of the center of the Gabor function, that is not necessarily the same as the center of the frame, since the filters can be slightly shifted. But most importantly, we get an orientation, is the orientation of the Gabor function. So the reason why we need to perform this change of coordinates is just to express the connectivity kernel in terms of relative positions and orientation and orientations of the filters. So to remap the kernel into the space of, into the rototranslation space. So the learned kernel that governs lateral connections in our architecture basically expresses the strength of interaction between two filters of indices f and g that are shifted in space by a vector ij. So this is a kernel that acts on the feature space of the layer as a sort of transition kernel, in the sense that it slightly modifies the activation of the layer based on the reciprocal interaction, the pairwise interactions between points of this layer. So this interaction can be positive or negative, meaning that the activation at one point will enhance, so increase the activation at the other point, and vice versa, or suppress the activation at the other point in case of negative values. And in case of zero values, that means that the activation at the 2 points just depends on the feed-forward input that they receive. So we want to express the same thing, but in terms of relative positions and orientations. So by performing this change of coordinates, we can reparameterize the kernel. So now this lives in R2 times S1. And for instance, for these couple of filters oriented vertically that you see here, if the original coordinates show a shift between the frames of the two filters of 3 times 5 pixels, this gets slightly modified, it becomes 0,4, and now it represents the shift between the centers of the Gabor functions. But most importantly, instead of fg, that are just two indices, we get the third coordinate, that is the difference between the orientations, that is 0 in this case, since they are oriented the same, or in this other case, it is, for instance, minus 45 degrees. That is the difference between the orientations of these two filters. So now we have a kernel that is expressed in terms of positions and orientations. We can study what kind of connectivity it induces. So for short-range connectivity, this means looking at basically the kernel where when the first two coordinates are 0, so no positional shift, but just the interactions between cells that lie at the same spatial position on the retina, but are sensitive to different orientations. So basically a one-dimensional function of orientation. And what we obtain, so these are the value of our learned kernel restricted to 0 positional shift up to a slight smoothing. And what we obtain is actually a inhibitory behavior for non-optimal orientation. So this is consistent with the non-maximal suppression mechanism that I was showing before. But then for long-range connectivity, so studying the long-range connectivity induced by the learned kernel means asking, given a filter psi f, which other filters, so at each other spatial position, which is the filter that interacts maximally with psi f. So that means taking the maximum for each spatial position, the maximum of the kernel over the orientation theta. So yes, maximizing the interaction between the two filters as a function of their orientation. So relative orientation. So the image here shows the projection of the kernel onto the XY plane obtained by taking this maximum. And the tiny segments that are superposed onto this represent for each point, the spatial, for each position, the orientation that maximizes the value of the kernel at that point. So in principle, there would be no reason to expect to obtain a vector field that has some sort of coherence in terms of orientation. But this is what we get. And most importantly, the vector field is aligned with the orientation of the starting filter, so that this tells us that each segment is a maximally interacting filter that we find at that point compared to the starting filter. So the maximally interacting filters are the ones that are either collinear or co-circular with this orientation of the starting filter. So I have no time to talk about this properly. I just wanted to give an idea of how to quantify this result by comparing it with existing theoretical models of connectivity. So in previous work, basically the spread of neural activity onto the cortical surface has been described in terms of a diffusion equation governed by a sub-Laplacian written in terms of these two vector fields in R2 times S1. So in this framework, one can also reobtain the field of reciprocal influence of locally oriented elements. So in other terms, the field that is called the association field, or in terms of interactions between filters, this is the field of orientations that are maximally connected with the starting orientation. So this can be obtained as a family of integral curves of these two vector fields projected down onto the XY plane. So in subsequent work, this has been shown to be also, so these curves have also been shown to approximate the statistics of co-occurrence of edges in a set of natural images. And with a similar approach, we compared these curves with the integral curves computed from the vector field that we obtained from maximization of the kernel at each point. And what we obtain is that these integral curves are actually closely approximated by those theoretical curves obtained from this established model of cortical connectivity. So I'm not going into any more details since I have no time. I guess that the take-home message is that carefully built computational blocks can develop geometric properties that are biologically relevant and that can also improve the performance of the network when they are trained onto natural images. So this is analogous to the plasticity of early visual pathways when exposed to the natural world. And the nice thing here is that almost all of the geometry is learned from the data, since the only constraint was the convolutional structure of the network. And of course, this is a first study, so there's a lot of room for improvement and extensions to, for instance, larger images to study a less local behavior, and also other analogies between deeper layers of the networks and higher, more complex areas of the cortex. So that was all. I just want to thank my co-authors and, of course, you for listening, for any comments or questions you may have.